Welcome to EduRev.
Welcome students. I'm Clarice Pereira Colasso and I have been training NEET medical aspirants in botany and zoology as well as NCERT based board exams for the past 8 years. Today in this video we will study the second chapter of our class 12 syllabus, and that is sexual reproduction in flowering plants. Now, sexual reproduction in flowering plants can be studied in three parts, that is pre-fertilization structures and events, followed by fertilization structures and events, and then you have post-fertilization structures and events. So if you look at it, today we will be studying in this video pre-fertilization structures and events, mainly with respect to the male reproductive part, that is the androecium, that is that part of the flower, that wall of the flower. So in this we will study the structure of the androecium, followed by microsporogenesis, that is the formation of the pollen grain, followed by microspore development, which is nothing but pollen grain development, so further development of the pollen grain. So to start off with the structures, pre-fertilization structures, we will focus on the structure of the androecium or the structure of the stamen. Now, technically speaking, a typical androecium consists of two different parts, that is the anther and the filament. Now, what is the difference between the anther and the filament? So if you look at the figure over here, the topmost portion, the projected portion, is the anther, okay, and the part that helps in projection is called as the filament. Now, there's a characteristic difference between these two. For example, the anther contains developing pollen grains within it. So we can call this as the fertile part of the androecium. However, the filament is simply an attachment module. Okay, so the filament is called as the sterile part. It only helps in providing nutrition to the developing pollen grains within the anther. Okay, so it draws nutrition from the flower below. So if you look at the whole thing, the stamen containing the anther is meant for pollen grain production or pollen grain development, but the filament is meant for attachment of the anther, okay, attachment of the androecium, and it provides nutrition to the developing anther on top, or in other words, the developing pollen grains within. Now, let's study the structure of a typical anther of an angiosperm. Okay, so we're dealing with flowering plants over here. Now, very characteristically, if you look at this anther, it seems like it has two lobes, one in the front face, so that is one anterior, and one posterior. So we can say that a typical angiosperm anther is nothing but a bilobed anther. But technically saying, if you take a cross section of this particular anther, within it you will find that every lobe, every single lobe contains two sacculated structures within it, so two sacs present within it. So if you say every lobe will have two sacs. Now the technical term for sac used here is theca. Okay, so the word theca actually means a casing, so a sac in other words. So if a typical anther is bilobed and if every lobe contains two sacs within it, I can definitely say that a typical angiosperm anther is tetrathecous. You have the presence of four theca present within it. Okay, so this term tetrathecous and bilobed is very, very essential for your NEET exam. So you should be able to know this. Okay. Now, if you look at the cross section here, very clearly you can see four sacculated structures, two on the left hand side, two on the right hand side. So in total, four sacculated structures. Okay. Now, these sacs very clearly contain pollen grains in them. So we can say that these four sacs that we were visualizing were nothing but the pollen sacs. So they contain pollen grains. Okay. So your tetrathecous condition is the condition of the pollen sac. Now we have a technical term for pollen sac. Pollen sac is called microsporangium. The word sporangium means sac. The word micro implies small. Now actually speaking, it's called small sac because the pollen grains are called microspores. They are the smaller gamete. Okay. Actually speaking, the pollen grain is not the gamete. It contains the gamete. So pollen grains are called microspores, but the pollen sac is called the microsporangium. Okay, it contains the microspores. Now, let's study a little bit about the microsporangium in depth. Okay, let's study a little bit about it. Now, the microsporangium consists of two parts. You can divide it into two parts. The outer wall layers. Now, these outer wall layers are characteristically four in number. Now, they have a number of different functions. Okay, and internally you have something called a sporogenous tissue, which is nothing but your meiocytic tissue. This is the tissue that undergoes meiosis. Okay, So let's deal with the first part, that is the wall layers. Now let's look at these four wall layers. The first wall layer is called as the epidermis. Okay, so it is a single layer on the outermost part. The second layer is called as the endothecium. Now this is also single layered. The third is the middle layers. Now these middle layers range between 1 to 3 depending upon the species that we're talking about. Okay, but characteristically speaking, these outer three layers out of the four, these outer three layers are basically meant for protection. And when it comes to the time of pollen grain release, which means dehiscence of the anther, it is very essential that these three dry up and therefore allow the pollen sac to basically open, the anther to dehisce, the anther to open up. But we're done only with three layers. These are protective in nature. But the fourth layer that we talk about is called as the tapetum. Now, this particular tapetum is the closest to the sporogenous tissue that we will study a little later. So, can we say in a way that this particular tapetum provides a lot of nutrition to the sporogenous tissues on the inside? Now, what is the function of the sporogenous tissue? Sporogenous cells are supposed to be myocytes, which means they will give rise to the pollen grain. So in other words, the tapetum's main function is to provide nutrition to the developing pollen grain on the inner side. So if we have an overlook of this, the four external wall layers, three of them being protective, okay, that is the epidermis, the endothecium and the middle layers. Finally, the nutrition providing layer is nothing but the tapetum. So let's look at the characteristics of the tapetum a little bit further. So that is what we're talking about. Tapetum very close to the sporogenous tissue, giving rise to the pollen grains. So the first characteristic of the tapetum is that it is nutritive. Now the fact that you are nutritive, very often you will find that nutritive cells are multi-nucleated, which means they have more amount of DNA. If you have more amount of DNA, you have the capacity to produce more amount of protein. So in other words, you will be able to secrete this protein to other cells. Okay, so you will find that also here, the tapetal cells are said to be multi-nucleated cells. Now, apart from being multinucleated cells, they're providing nutrition to the developing pollen grains on the inside. So they form a number of components for those particular pollen grains. For example, they give pollen grains a particular compound called as sporopollenin. Now, sporopollenin is actually meant to form the outermost coating of the pollen grain, and it is a very, very resistant material. But this material is synthesized by the tapetum. Likewise, in some species, not all, the tapetum will also provide an additional layer, and that is called as, or rather an additional material, and that material is called pollen kit. Now pollen kit is a yellow sticky material, so yellow, therefore you can understand it's made of carotenoids, okay, and it's a sticky material. Now definitely pollen grain that require insect pollination need to be sticky in nature so that they can stick onto the pollinating agent, okay. So you will find sporopollenin is the outermost covering of the pollen grain provided by the tapetum, and the second is pollen kit is the sticky surface present on a pollen grain. So only if you require insect pollination will the tapetum secrete this particular entity. Now, so we said that the microsporangium has four wall layers. Now let's deal with the second part of the microsporangium, and that is the sporogenous tissue. Now this particular sporogenous tissue is myocytic, which means it's a diploid tissue, but this particular tissue is able, or it is subjected to meiosis. Okay, so with this we are done with the pre-fertilization structures of the androecium, and now we will start focusing on the pre-fertilization event of the androecium. Okay, so what really happens to these particular myocytes? Now these myocytes enter a process called as microsporogenesis. Now microsporogenesis basically means the development of microspores. Okay, so development of microspores, or the formation rather of microspores, means the formation of pollen grains. Now in order to form these pollen grains, you require simply one process to occur, and that is a single meiotic division. That is all that is required. Okay, now your myocyte or your sporogenous tissue have a whole lot of other terms which we will come to very soon. Another term for the myocyte is definitely the microspore mother cell, and we can understand why so. Pollen grains are called microspores, so therefore the myocyte that forms it is called the microspore mother cell. Remember, this particular cell has to be diploid in nature because we're talking about a higher plant. Okay, we're talking about angiosperms. These are generally diploid. They have diplontic life cycle. So we have studied this in previous videos. So let's go through microsporogenesis in depth. So microsporogenesis starts off with a tissue called as the sporogenous tissue, okay? But every cell of the sporogenous tissue, now mark my words here, every cell of the sporogenous tissue is capable of forming a microspore or capable of undergoing microsporogenesis. So every cell has the potential to form microspores. Therefore, another word for sporogenous tissue are called potential mother cells. So in other words, PMC. But this word PMC can also be given for pollen mother cell. Okay, so two ways. And you can also call it as the microspore mother cell. So definitely this diploid cell will undergo meiosis 1, which is reductional division, to give rise to two cells. So this has to be a microspore dyad. These are going to be haploid in nature. But remember, meiosis has not been completed yet. So you need to undergo meiosis 2, which is equational division, and you will result in the microspore tetrad. So you have resulted in 4 pollen grains, all of them haploid. However, we must remember at this point that these particular microspores that are formed in the tetrad are very, very immature and they must undergo further development. So the next part of pre-fertilization events would deal with the development of those particular microspores. But to understand the development well, let's look at the structure first of the microspore of the pollen grain. Now please remember, the microspore or the pollen grain is not really the male gamete, okay, but rather later on you'll understand that it houses the male gamete. So if you look at this particular structure, every pollen grain has different shapes depending upon the type of species and the method of pollination later on. But generally speaking, they have a diameter which ranges between 25 to 50 micrometer. Now, this particular number is very frequently asked for your NEET exam, so kindly study it. It's very important for you. Okay, but if you look at this particular pollen grain, it seems to have two wall layers, two outer protective walls. Okay, the outermost wall is called as the exine, the inner wall is called as the intine. So this is what makes up the wall layers. Now let's look at both of these individually, starting off with the exine. Now if you look at the exine, we said that the tapetum secretes sporopollenin, helping to form this particular exine. But sporopollenin is a very, very resistant material. It's the most resistant organic material. Now why do we say so? We say this simply because there is no known acid, no known alkali that can destroy it. There's no known enzyme for that matter that can destroy it. And even at higher temperatures, it seems to remain intact. It does not break down. Okay, so it seems to be very, very resistant. Now, how is that helpful for us? Okay, or how is that helpful to the plant? We can say that this helps to increase the viability of pollen grains, and for that matter, we are able to find pollen grains that are viable over many, many years, thousands of years. They remain viable even in fossils. Why? Simply because of the presence of pollen grain in the exine. But we have a problem. Later on, we need a pollen tube to germinate from the inside of the pollen grain. Okay, from the inside to the outside. But if the exine is going to be so resistant, if the exine is going to be so hard, it's going to be difficult for this particular pollen tube to generate out, because we said there is no known enzyme that can break it. So therefore, the exine has some holes here and there, basically 1 to 3 in number, called as germ pores. And these germ pores are specific areas in the exine that do not have sporopollenin. So definitely things can, or material can ooze out of it, or in other words, a pollen tube can generate out of it. Okay, so that's the germ pore. Now, coming to the second part, that's the intine. Now, the intine is a very thin layer, okay, compared to the exine, very thin layer, stretchy layer. Okay, so it is made up of cellulose and it is made up of pectin. These are living materials when we talk about the plant kingdom. Okay, so this is what your intine is made up of. Now, internally to the intine, you will find the protoplasm, so the living part of the cell containing a lot of food, containing the nucleus. Now, let's go into further development of the microspore. So we said microspore development. Okay, now microspore development involves two different stages. The first is interphase, followed by two mitotic divisions. So there are two subsequent mitotic divisions. Now, what happens during interphase? We already know that interphase is a period in which you increase the bulk of your cytoplasm. So that's exactly what takes place in the pollen grain here. So you find that the food reserves keep on doubling. So you can see a number of food vacuoles over there. Okay. Now the moment this interphase is over and the pollen grain has increased its cytoplasmic material, it will enter the first mitotic division. Now this mitotic division is unequal, as you can see, and the pollen grain seems to generate two cells, a vegetative cell and a generative cell. Now though we seem to draw the vegetative cell on top and the generative cell below, you need to understand that the generative cell is actually floating in the cytoplasm of the vegetative. It is not separated, it is floating in the cytoplasm of the vegetative. Okay. Now, definitely speaking, this is a two-celled stage. Okay, the pollen grain seems to be in a two-cell stage. Now, up to this particular point, development took place in the flower on the plant. Now, most angiosperms, around 60% of angiosperms, will have anther dehiscence and pollen grain release at this particular stage. Okay, the remaining 40% wait for a little bit of time, but 60% will release their pollen grain at this particular stage. Now, let's try to understand the functions of these individual cells, starting with the vegetative cell. Okay, now the vegetative cell is nutritive. It has a lot of the nutrition, most of the cytoplasm is present there. And later on you will find that this particular cell gives rise to the pollen tube, okay, which is very essential, gives rise to the pollen tube. And the generative cell. Now, this particular generative cell is the one that undergoes the second mitotic division. So, if it undergoes the second mitotic division, we can clearly say that the generative cell will give rise to two cells. So now, technically speaking, the pollen grain has one vegetative cell, but it has two daughter cells of the generative cell, which means now the pollen grain is in the three-cell stage. So the remaining 40% of your angiosperms will undergo anther dehiscence at this particular stage, three-cell stage. Now, what are the two cells that the generative cell gives rise to? So the generative cell gives rise to the two male gametes. So this is what we need to be careful of. The pollen grain is not really the male gamete, but the pollen grain houses the male gamete. Okay, so this is what we must remember: the two-cell stage and the three-cell stage. So let's go into a couple of MCQs to help you out with your NEET exam. Okay, the first one: how many of the following statements are correct with respect to pollen grains? Now you have a couple of options we need to choose. The first option: pollen grain is the male gametophyte. Now the word gametophyte means the housing structure. Okay, now the pollen grain is the male gametophyte. Now that is a true statement. It is the male gametophyte. Sporopollenin is made up of carbohydrates. Now that is false. Intine is made of carotenoids. That is also false, simply because the intine is not yellow in color. The exine can be yellow in color. Pollen kit is produced by tapetal cells. Now that is true. Okay. Pollen grains do not depend on temperature and humidity to remain viable due to the presence of sporopollenin. That's true, because sporopollenin is resistant. The next: pollen shows precocious germination. A precocious germination implies germination within the parent, germination when it is still young. Okay, now why do we say germination? When we say a microspore develops into a mature pollen grain, okay, that is called precocious development. So that is also a true statement. So three statements are true. So the answer here is option B. Three are correct. Okay, let's go on to the next question. Which one is correct about a pollen mother cell? So the meiocyte. Pollen mother cell is a pollen grain. Definitely not, but it gives rise to one. Each cell of the microspore tetrad is a potential pollen mother cell. No, that's not right. Third one: each cell of the sporogenous tissue is a potential mother cell. Now this is correct. Okay, so this is a correct statement. Pollen mother cell is diploid. Definitely, it's a meiocyte. So this is also correct. So therefore, if you go through the options, the answer here is option D, which says that the third and the fourth statement are correct. So students, I thank you for attending today's video. I'm sure you have understood a lot about the pre-fertilization structures and events of the androecium. I welcome you to go through EduRev Infinity, where you will come across a whole lot of other videos. It will help you prepare for your NEET exam, along with a lot of tests and a lot of material for you to read through. And I'm sure by going through all of this, you will increase your nationwide ranking. Thank you, students.